I want to explain why living things have a Latin sounding name and a little bit about the history and the background. Hi, hello and welcome, Microbehunter here. Well, I'm sure that you've already heard or that you've already read somewhere that living things are given a scientific name which is made of two parts. Yeah? So I want to give you an example here. We all know apple trees, right? And apple tree, that is the so-called the trivial name, the name of the plant that we use in everyday life. But there is a scientific name as well called Malus domestica. Malus domestica, that is the apple tree. I'll give you a second example. What about us human beings? We humans, the word human, that is the so-called the trivial name, but the scientific name is called Homo sapiens. And the blue whale has the name of Balionoptera musculus. Wow, that's a long one. And the fruit fly, it's a small fly, Drosophila melanogaster. So the question is why is that? So in this episode, I want to talk a little bit about this and the history and how it actually also changed over the years and some problems here. And what I'm going to do now is I'm going to turn the time back a few hundred years, into the 1700s. This was the time when in the Western world, at least, a new movement started called the movement of enlightenment. This is the time when people started to think rationally, or at least a little bit more rationally about their environment. This was also the time of the Industrial Revolution in Great Britain, which then spread across the world. And at that time, scientists started also to collect a lot of organisms and plants and animals from around the world. And they also started to classify them and to group them and to categorize them. And it should not be surprising that for many of the new living things that they found, they didn't have a name. It's not only that they were new discoveries, but sometimes the plants simply and animals were not local, and therefore, there was actually no name. I mean, apple tree is a name that we're using in the English language because we know what apples are, and that's the fruits that we use. But what about the name of fruits and vegetables and plants that are not local to our place where we live? I mean, then we don't have a name, right? So this was one of the problems that the scientists encountered at that time. They went on expeditions, found new animals, found new plants, found new things, and they then wanted to give it a name. And what they have done, and this was actually a pretty smart idea, is they said, okay, we're going to use Latin. We have the language Latin, and we're not going to just give it a name, but the name should be very descriptive as well, and it should actually describe how the organism looks like. So what they've done is they have given a long description of, so that people who read this name actually also are able to then understand and picture what the characteristics are. Why did they use Latin? Well, I think, that could be one reason. You might do the research yourself on this, but I think because it's a neutral language, because at that time there was also a lot of scientific nationalism going on. And can you imagine now if French scientists would have given it a French name, then the German scientist would not have accepted it, and vice versa. If a German scientist would have given it a German name, then the French scientific community would not have accepted it, I would guess. Because there was between those two countries at that time also some kind of a scientific nationalism and competition going on. So I think having a Latin as a language of science is actually a good thing because it's a little bit neutral. But let's go back a little bit to the naming of this new organism that they found. I want to give you an example here, and you please apologize my bad pronunciation here because I do not know Latin, so I'm just going to read it off here. That's basically the name of a plant, and just look, this is the way it goes. Plantagofolis ovato lanceolatus pubescentibus spica cylindrica scapotereti. That is the name of the plant. Okay, I'm going to translate this into English. It's a plant with pubescent ovate lanceolate leaves and a cylindrical spike and terete scape. What in the world does this even mean in English? I mean, I'm already having problems understanding it in English alone because it's full of botanical terminology. So it makes a lot of sense to maybe to a botanist who understands this, but I mean there are a whole bunch of disadvantages here. It's very, very impractical to name a plant like this. And by the way, this plant is nowadays called Plantago media, and that is now the two-name system that we're using these days. Now, it was like this that back 300 years ago, it became evident quite quickly that names that are, I don't know, a line long, these are so-called the polynomial names, that's very impractical, and for one reason, because as the scientific discovery started to build up, they discovered that maybe there are also other plants that have those characteristics. And then what? And then you actually have to modify the description here and change the name around, and simply to accommodate for the differences to the plant that they just have discovered. And that's a problem. And a Swedish botanist, his name was Carl Linnaeus, he said that that's not practical. It's way too long. And what he has done is the following. He just cut it down to 2 words. He said that, okay, all living things have to have 2 names or 2 words. Like, for example, in this case, Plantago media. Or like the examples that I've given you before in the case of the apple tree, Malus domestica, 2 names, or human Homo sapiens. Fruit fly, Drosophila melanogaster. Always 2 names. And because at that time they realized the following, surprisingly, they realized, well, actually the description is not so important. What is really important is that the name is very unique. The name must be unique, because if you've got some kind of a reference book and if you've got a unique name, you can always consult the reference book for a full description anyway. But it's really important that the name doesn't change. But that's the problem that they had before. They made new discoveries and had to change the name around because they needed to adapt the description to distinguish, let's say, between 2 plants that look very similar. So it was very impractical, and so when Carl Linnaeus introduced the so-called the binomial system, the 2-name system, then this was readily adopted by many people because it had a whole bunch of advantages, which I would like to explain to you now. First of all, it is called the economy of words. I mean, when you write a scientific publication and when you have to describe a plant in 1 line or even in more than 1 line, this takes a lot of space and is not practical, right? So it's the economy of words. The second thing is the following, that this binomial, the 2-name system, is short enough that you can also use it in everyday language. I want to give you the example, Homo sapiens. I mean, that's a term that we use quite frequently these days, or even more, boa constrictor. That's a snake. We say, oh, it's a boa constrictor. So we see that we can use this even in everyday language, or maybe even more famous, Tyrannosaurus rex, the dinosaur. So here too we have the 2-name system in place, and it's so popular that we do not even have a so-called trivial name for those, like the Tyrannosaurus rex, or the boa constrictor. We just use even those words, the scientific name, in everyday English. It's that practical. So the widespread usability of those words is very good. Then the uniqueness, I already mentioned this, the uniqueness of the name. And then also there is another interesting characteristic, this is called the name stability. And this means that if all of a sudden you discover that it is necessary to rename it, then it is possible to change the first of the 2 words. And I'm going to explain this a little bit, because the first of the 2 words, like boa constrictor, boa, that is called the genus name. Okay, the plural would be genera. Or in the case of Homo sapiens, Homo is the genus name, and we belong to the species Homo sapiens. So for the species, you have to use both names together. Okay, or the apple tree that I mentioned before, the genus name is Malus, and the apple tree is Malus domestica. And if there is now the necessity to rename it because there has been a mistake, then what you can do is you simply leave the second part of the name and you change around the genus name, and then it's still evident that there has been a change here. It's a little bit like this. I know this is a strange comparison a little bit, but when a person marries and changes the family name, the first name still stays the same, right? So a little bit, this is an analogy, but you still are more easily able to identify the person. So this is essentially the advantages of the binomial system, and I want to give you a couple of examples here more examples here of different species within one genus. So let's talk about the boa constrictor, the snake again. Boa constrictor is only one species. There's also a boa imperator, a boa nebulosa, a boa sigma. Okay, all of them are different species of the genus boa. Yeah. So they are highly related, not highly, or they're reasonably related, but they're different species. That means they're not able to reproduce with each other. So a boa constrictor is not able to reproduce with a boa imperator, for example. Okay, this is actually also the definition of a species. So you see, it's pretty practical to use this two-name system here. Maybe you've also heard of the bacterium E. coli. E. coli, that is an abbreviation because the E stands for Escherichia, but there are also a whole bunch of other species that belong to this genus. For example, E. albertii, E. fergusonii. I think they must have been named according to someone with the name of Albert and Ferguson. But you see that the name here does not always necessarily now have to also be descriptive of the organism. It can be also named in honor of a scientist. And that's possible as long as the name is unique. Okay? Because we're not anymore bound to the description, because as I mentioned before, all you got to do is consult a reference book if you want to know how the organism is. Yeah. So this was a change in thinking of the function of a name, really. You move away from a description towards uniqueness. And this is essentially also something that we are using these days. Now, when you are using a scientific name somewhere, then you should stick to a few conventions and a few regulations here, which is also internationally standardized. In printed text, the name must be italicized, so it must be slanted, and the genus name, that's the first one, must be capitalized, and the second word must be in lowercase letters. This is international, an international standard. Now, what do you do actually when you're handwriting it? When you're handwriting the name of organisms, well, in this case, you have to, of course, also capitalize the first word, and the second one must be in lowercase. But then you also have to underline it. Yeah, so even that is standardized, and has been used now for a long time. Now, there is, of course, a problem, and I would like to talk now a little bit about my own personal experience with this, because while I was still at university and writing my master's thesis, my topic was about in bacteriology, and systematic bacteriology is basically the classification and identification of bacteria, using a whole range of DNA tests and biochemical tests. And this was pretty difficult because I got a whole bunch of bacteria. I had to put them into pure culture, and I had to do a whole range of biochemical tests on them, of course, do a little bit of microscopic characterization as well. But the microscope does not provide sufficient information to identify bacteria. And so what I had to do over a long time, I had to really dig through a lot of publications and a lot of papers and scientific tables and data tables and so on, because historically some bacteria have been dumped together even though they were not related. And I want to give you an example here. In the case of the genus Pseudomonas, that's the name of a genus, and this was actually the genus where people who have identified bacteria that were staining gram-negative, so that means under the microscope they had a red color because of the staining reaction. They were long and rod-shaped, so they were kind of elongated, and they had, on one end, they had a flagellum, which is a structure that rotates very quickly to move the bacterium along. And if a bacterium had those characteristics, then the scientists said, it's a Pseudomonas. Okay, you dump them together into the category, into the genus Pseudomonas. But the problem was they were sometimes totally unrelated. Because in the field of biology, what we want to do is we want to group organisms together based on their so-called phylogenetic or evolutionary relationship. Okay, so to go back to the example of the boa, of the snake, boa constrictor, boa imperator, and so on, boa sigma, all are genetically similar and they have a common ancestor because they're all boas. Okay, and it doesn't matter what color they have and what size they have. Yeah, of course they will look mostly similar because they're related. Yeah, but it doesn't have to be. And especially with bacteria, it's a huge problem because these characteristics here, the way that they stain, what the shape that they have, and whether they have a flagellum to push them forward, honestly, that is so general that you can find them in so many also unrelated bacteria that these are not really good characteristics. And to give you an analogy, it's almost like saying, okay, all blue cars that I have are Mercedes, all green cars are BMWs, and all white cars are Fords. Yeah, of course you're not. You're going to say, I mean, color is not a criterion that you should use. Yeah, we all understand this. You cannot simply say if a car is blue, then it's a Mercedes. Of course you cannot say this. We all understand this. But this is exactly what they have done with bacteria. Yeah? Because they didn't know better, because what they've done is they used a microscope and they were able to see those shapes and those structures and those colors of the staining reactions. And then they say, okay, these are the characteristics, they must be related, and let's dump them together into the genus Pseudomonas. And this happened a whole bunch of times, many times. Yeah? And we now know that this is not enough, and that we actually have to do DNA studies, or we have to do a whole range of chemical or metabolic tests to distinguish them much better. Because organisms, we now know, that are highly related can look different, or at least bacteria, can look different under the microscope and vice versa. Yeah? Very distantly related organisms can look very similar, okay? And just because two bacteria have the same shape does not mean that they're related. They can be very different. So there is all of these possibilities exist. And for this reason, microscopes alone are not enough to identify bacteria down to sufficiently high degree. Of course, I can see them and say, these are bacteria. Of course I know that. But which ones they are, I mean, I don't know. It's almost like saying I look at an organism, it's an animal, or it's a plant. Yeah? But it's not enough for me to distinguish them further just by using a microscope. Yeah? So this is a little bit one of some of the issues that we're having here, and which makes taxonomy and classification and identification a little bit difficult, is even though we want to have this name stability, which is so important, even that is not always the case, okay? So because so many of the organisms have been dumped together into one genus, even though they're not related, scientists had to do a lot of cleanup work in the last couple of years, and there was a lot of reclassification and reassignment, and it's a highly dynamic system and a highly dynamic thing, and which makes life a little bit difficult for like a student like that I was, because I had to kind of trace back a little bit is how were the names of the different bacteria, how did they change actually over the years? Because scientists made new discoveries and then they published a paper, well actually it's not a Pseudomonas, but it's a different one and so on. Yeah? And to make things worse even, especially with bacteria that are medically important, you cannot rename them anymore, because in medical literature, it's so established that if you were to rename them, it's just going to cause a lot of confusion. Give you an example here, that some scientists actually say that Shigella, the bacterium with the genus Shigella, and Salmonella, many of them are really actually so related that they should be the same. Yeah. But you cannot simply rename them now, because in medical literature we just use those terms, and we cannot just say, okay, we're just going to change the name around. Yeah, you can't do that. Yeah, it's just going to cause confusion, and so scientists or the community simply decide to sometimes leave the names as they are, because they are already so established. Yeah. And you cannot just simply turn around and then and say, we're just going to rename it, and it's just going to cause even more confusion here, right? So you see that the whole thing is a complicated issue here, and because it's so fun, I'm going to even complicate it yet more, because I just talked about how we can have gone from a trivial naming system like apple tree to to a binomial system like Malus domestica, and now I'm telling you, well, actually, it's possible to have three names even. 3 names. Can you believe this? Yeah. Give you an example here. The family dog, right? The domestic dog is Canis lupus. Canis lupus actually is also the wolf, because the domestic dog that we have, the family dog, that we have is actually a wolf. Yeah, it's a product of the wolf. But I think we all understand now that a wolf and, let's say, a small dog that we have, I mean, they're not able to reproduce with each other. So what scientists then said is, well, actually what we have to do is, they're actually still the same species, because theoretically they could reproduce genetically, but actually we should make a distinction here. So what they've added, sometimes they added is so-called a subspecies as a third name. To give you an example here is Canis lupus familiaris. That is the domestic dog. Yeah? And because it's so popular now, Canis lupus familiaris, there's so many domestic dogs nowadays, what they say is, well, you know what, let's make a separate species out of them. Let's just call it Canis familiaris, because it's not really possible that they interbreed with a wolf anyway anymore. Huh? So there are sometimes even synonyms here. Okay, that sometimes multiple names actually refer to the same species. And this can also be the case if, for example, scientists in different countries, when they actually give a different name to the same organism. This can also cause confusions, and this means that for some organisms there are also synonyms. There are multiple names possible. Even that isn't a thing here. And this can really lead to a couple of crazy names here. For example, the Western lowland gorilla has the species gorilla. First gorilla is the genus name. Gorilla is the species name. But because it's the Western lowland gorilla, you have to add a subspecies, and they call it the subspecies, you guessed it, gorilla. So the Western lowland gorilla is called gorilla. To contrast it from the Cross River gorilla, which has the name gorilla dieli. Okay? So the Western lowland gorilla and the Cross River gorilla are the same species, gorilla, but you kind of still distinguish them a little bit from each other because they're different subspecies, and that's why you add a third name here. Or for example, the human being, we modern humans, we're called Homo sapiens sapiens. By the way, it's a pure coincidence that the third name is a repetition. Yeah, or an extinct Homo sapiens was called Homo sapiens neanderthalensis. It was the Neanderthal. Yeah, same species as we are, but a different subspecies. So, and if we stay with the human, I mean, our genus is Homo. Are there any other species in this genus? And actually, they are not. Okay, in the case of human beings. Yeah, we are the only species in this genus. Yeah, it's a little bit semi-arbitrary where you draw the lines, but I think we always consider ourselves as humans a little bit special and separate from nature anyway. I'm mentioning this with a slightly critical undertone, as you might have guessed. Yeah, so you see that this feeling of being special also reflects in the name that that scientists have given ourselves here. So yeah, in any case, this is basically all I wanted to say here, to give you a small insight into the naming of living organisms. This is quite a fascinating topic, but also a somewhat arbitrary topic. Yeah, because it carries a big historical load as well. And also, I would say, the high dynamic nature of science. There are new discoveries, and then you try to have a naming system that kind of keeps up with that. Yeah, also adds a little bit to the complications. All of this naming has a very practical implication for microscopy as well. I need to talk about this here as well. When you see an organism under the microscope, especially a protozoan, for example, or a microanimal like rotifers. These are multicellular animals, and you would like to identify them. There are lots of people who ask me, okay, how do I do that? Well, I give you a suggestion. What you do is you buy yourself an identification book, which contains a lot of drawings. And what you do then is you try to match this image that you see under the microscope with the drawing that you have in the identification book. And now an identification book is never able to contain all of the species because the biodiversity is simply too large. But maybe you're able to find an organism that looks very similar to it, and then you have a name for this organism. Because it will actually tell you next to the drawing, it will tell you the scientific binomial name. And then, if you have a name, then you go into Google, you type it into the search engine, and then you're able to see some pictures, some photographs that other people made, and then you can actually compare your organism that you found under the microscope with the picture that you've got from the search engine. And you're going to find something very surprising, that it's sometimes very difficult to find organisms, to find a match. Sometimes there is a high degree of similarity, but some differences, and these differences do not mean that you have got a different species now, but it could also mean that even in the same species, because of different growth conditions, that they develop differently. Okay, so you see even here, this requires a little bit of an experience so that you're able to evaluate that, what you're able to see, and to interpret that, what you're able to see. Yeah? And if you're specialized then, and if you dig in deeper, and if you're specialized with certain groups of organisms, then it's actually much easier, because then you know what to look for. And then still, you will probably discover that no reference manual or no identification book is able to cover all of the species, because, believe it or not, many of the things have not yet been discovered, and the chances are there that you might actually see a species under the microscope that has not yet been described. But we don't know that, okay? So that's quite a possibility as well. Yeah? So I think I'm finished. I hope that it was a little bit informative for you, all of these naming things concerning living organisms. I wish you all the best. Happy microbe hunting as always, and see you around next time. Bye-bye.